I'd like to present Dr. Samarendra Mohanty.
Good morning, everyone. So, thanks for coming to my lecture. So, I'll be presenting light-assisted construction, control, and understanding of neural circuitry. Okay. So, a lot of data that I'll be presenting are first time you will be seeing coming out fresh from my lab. Even some of them are not published. Okay. So, you are the first in the world to see those. Okay. So, what my lab is working on, as you see, not only on in vivo, but also at the in vitro level. Okay. So, in the in vivo level, we are using light to control different functions. But for today's talk, I will restrict myself only to in vitro, because all things in the world comes to the micro and nano level, lot of things can be changed. For example, if you see in the medical sciences, a microscope which can see bacteria, a small structure, could lead to development of lot of medical products or therapeutics. So today we will be focusing primarily on in vitro. In vitro means at the microscopic scale, what is going on. Okay, so as you see, the brain is very complex. I could not draw all 100 billion neurons to fit into this resolution of the screen, but I have drawn few neurons. Okay, and those neurons make connections with each other, trillions of connections. And we are only talking about now brain, but if you consider the whole nervous system, then there are so many more trillions of connections all around your body. Okay, so it's very complex. So there is a brain initiative now taken up by both US and Europe, but there are two different approaches to ensure how these connections are existing. Is it physical connections? Also, is there a functional connection? So to map those, in US, the brain initiative only wants to see mostly the functional aspect of it at the whole brain level, but none of the existing imaging technology, whether it is functional MRI or optical methods, can image the brain with single cell resolution of these 100 billion neurons. In fact, they can only see millimeter by millimeter cube using optics. Okay, so you cannot go deep into the brain by using optics, and MRI does not provide you that resolution. So it's like thousands of order magnitude big task to understand the whole brain. Okay, and in the European initiative, they are simulating this 100 billion neuron, one neuron by one neuron. And to simulate one neuron, you require one laptop. So you can imagine 100 billion neuron will require almost 100 billion laptops, which is thousands of times bigger computing power than the supercomputer that exists today, okay, by IBM. So it's again, there has to be very innovative technology by which computers have to be developed to understand brain, okay, because brain is so complex. And that's why, as you see, the brain only uses 20 watts of power to do computation called cognitive computation. Okay, means we make decisions, we generate knowledge based on the knowledge, whereas this computer does not do that. You understand? So that cognitive function is very, very important. And what is now being understood, we also hypothesize, or we want to find out how many neurons are sufficient to generate this cognitive function. Do you require a big brain, or there are animals which has only few hundred neurons, they can generate cognitive function, this decision making. So since there are small animals which can do this, so we believe if we go to the in vitro level and we build these neurons or brain circuit with few hundred neurons or even less than that, so we want to determine that which will generate this cognitive function or cognitive computation. And that one will help in ultimately to develop most probably a bigger supercomputer which can be applied to understand the brain. Okay, so as I told you, brain is very complex, as Emerson Cox said. If the brain would have been so simple that we can understand it, then we could not. Okay, so it's really very difficult, challenging task. Okay, I already described about how brain is consisting of billions of neurons, trillions of connections, and they're responsible for all functions. So it's not just the genomics, but the connectomics, functional connectomics of this brain, which defines each of us, how we behave, how we think. Okay, so it's very, very important how the brain, the cells, neurons are connected. I'm not going to go through this detail. I already talked about this. So we want to see this cognitive function. Okay, there are some key questions that we are asking. What is the information content of this neural circuit? Okay, how do you define information? As some people have recently discovered, if you see a driver, cab driver in London has lot of physical connection in particular part of the brain, because he has to remember all the lanes and everything. Okay, but is it only about physical connections, or is it about expressing some biochemicals on the surfaces, or it's a convolution of those two with the electrical activities? He might have big connection. If he's not using it, or there are not enough electrical activity, we believe that may not contain the information that brain requires to process if a situation arises. So our lab is now focusing, this is one of the major activities, that how to define information content of a network, how to develop from experimental measurements that I am going to show you, from well-defined circuits, how to define this information content. Once we define that, how the information content changes after potentiation. Potentiation means you practice something again and again, same way we can encode by light, stimulate and encode this information into the system. Can we do that? And how that flows through neuronal network, and what is the role of different pathways for this net output that we generate, the cognitive function. And as I told you, how many number of neurons are sufficient to generate this cognitive function? 4, 5, 6, 10, 100, like that. Okay? And if we cause an injury in this circuit, then how this circuit map, the functions and connectivity will change, that we want to find out. And how then neuron circuit process information. Then you can have plethora of analysis. For example, how the drugs will change these things. Okay, so you can analyze lot of, it can have lot of biomedical application, and also for developing, as I told you, computational tools. So our lab is focused now for all optically controlled or optically constructed neuronal circuit. So I hope you understand why we are doing this. So what is the tool we will be using? So I am going over each of them. Okay, so lots of results I cannot describe which are in high impact journal, but I will try to present you whatever is almost accepted, okay, or in minor revision. Okay, so we will be using several technologies that we have worked over past decade. So we think we are uniquely poised not to ask these questions, but also we have these technologies in hand. One is multiple beam optical tweezers. Okay, these are like laser tracking beam you see. So you can hold neurons derived from brain, I'll show you, and then you can position them however you want. Let's say in this topological structure, a triangular or a linear arrange of this, or in a cube like this. Not only that, as you know, these neurons are different color, different sizes. They are, let's say, from different parts of the brain, and you want to organize them in a particular fashion. So we hold by laser beam these individual neurons and make the circuit in a three-dimensional way. Okay. Then the most important one is, once they are in place, how to make the connections between them. That is the most important part, as I told you. The connection has to be there. Just having neurons having no connections, information cannot pass through. You have to have the wire connecting. Okay. So these are the wires called axons. So we'll show you how light is used to guide the neurons to make them connect or disconnect. Okay. So this laser scissor is a nanosurgical tool where you can dissect this. I'm going to show you that you suppose do not require this connection, you can cut it. Okay. And then if you want to functionalize some of these neurons, functionalize means give some particular genes to them, give some particular chemicals to them, so that they have some, basically change the information in the circuit, then we are using laser to deliver the genes or the molecules that is required. Okay. And then comes light in combination with genetics. If we shine light, let's say you construct a circuit like that, and then I shine light, all white light, everything will get stimulated. But we want specifically particular cells to be encoded with some information. Information by means, if I give pulses of light, I can make that neuron to fire, synchronized to my light. Okay, but to make that, there is a very, if you want to genetically target that to a specific group of cells, then we do something called optogenetic stimulation. Then I will come, once we have this circuit which we have manipulated, controlled by light, we want to record the activity. So we want to record activity not only electrical but also optical, because electrical is very difficult to poke into all different cells. So is there a way by which light can be used to detect neural activity? Okay? So measuring communication. So these are different tools I am going to go over. So one is a classical technology where what we are doing is, let's say you have a neuron has dendrites and long axons. These are processes which connect. The axon goes and connect to dendrites. Okay? So what we are doing by different technology, we are confining the neuron body, the cell body, to a small circle or a circle of 10 to 15 micron, so that the dendrites cannot migrate to distant neuron. Okay? So this is done chemically. Okay? Or physical LTL. Then once we guide it to this path by light, it can go and connect to the other neuron. So the strategy is to make this kind of chemically modified surfaces, okay, or physically modified, as I am going to describe that. So there are a lot of different ways that we are doing it. Then, as I told you, so there are different parts of the brain. Let's say visual cortex, auditory cortex, which is responsible for hearing, or somatosensory cortex for sensory functions, or prefrontal cortex for a lot of cognitive function. So we derive those neurons and then position them in this circuit that we made. Okay? And how we position them? We take with help of optical tweezers. So they are derived from the brain and put into a chamber, and then you take one cell at a time using light beam, tracking beam. Okay, I'm going to show you how that works. And then you position it how you want. Okay? How you want, you position them here, like you see here. The cells are being positioned in triangular fashion. I will show you some videos of that. So there are different types of neuron. You see some neurons, like you see the green ones, they are excitatory, and some are inhibitory. Okay, interneurons, like you see the red ones. So you can identify them by fluorescence. You can target them, you can identify them. So you can see here how optical tweezer can take cells or anything through one chamber to another chamber. That's what you are seeing. You mark this circle here, or the bright dot. That's an object being transported into neuron's circuit in the right side through light. So this you can do in a closed environment. So there is no contamination, light goes inside and hold things. And you can see more beautiful transport where you don't have to move anything, but objects automatically get moved from one point to another, okay, by light. And this is all inside enclosed object. How it works? It works by the force of light. This light falling on us is putting some pressure on us. We don't feel it, it's very less. But on the cells, molecules, small objects, the force is large enough to move objects. So what is happening, you can imagine light is focused, and light when interacts with a cell, let's say neuron, light bends, light does refraction, everyone knows that, isn't it? Reflection and refraction. So light when reflect or refract, it change its direction or momentum, and because of that, the object which is interacting comes, because of Newton's third law, every action has equal opposite reaction, so it will come back to the center of the beam. Basically light has force, and since light can be put into a large distance, you can manipulate it in a closed environment. So you can manipulate things inside cell. I am going to show you. But you can see how you can organize object. Here you are seeing 3 objects are being put there, and this is a 4th one, and we are releasing it. So you can manipulate and make this array of cells like you see here. These are neurons derived from the brain. This is like a rabbit paw structure, this is a line chain structure, this is triangular structure. So you can arrange, these are neurons derived from cortex of the brain, and you can see a video here how they are done. See here, one by one you bring this chain, you form, and you form this structures. So you can use multiple beams to arrange this, hundreds of these neurons, okay, in 2 dimensions or 3 dimensions, okay. But now the question is how they will connect this, how to guide them. So most of the methods are chemically oriented. Till now, all the research in neuroscience is to guide the neuron to to form circuits, you have to guide the axon. As I told you, axon is a process which connects to other neurons. So till now everything is mainly chemical based, okay? But chemical cannot control with this precision. You just apply, let's say your nerve is cut, you apply chemical and then nerve starts to grow, okay? So that is one technological question, that how we can do it non-chemically just by using light, because chemical you cannot, once you put it, you cannot take it out, you cannot have precise control, how to control these processes of neurons to connect, okay? But the more fundamental question is, till now people did not know that physical quantities, physical cues can also control neurons. That's a more fundamental discovery that we are making. So what is, just to give you a nomenclature of what is an axon, what is a neuron. Neuron has cell body, like as every neuron has its brain. Okay, like as you know, every cell has its own brain inside nucleus. It does all programming. It's a very complex structure. Every cell, even not neuron. So neuron is a cell. It has its own nucleus. But on top of that, neuron has a elongated hand-like structure. Like we have hand, so there it has one hand, like a long hand. It can be meters. It can be very small. Okay, it can be very long process, like a hand. So that hand has fingers, like you see there. Okay, so that finger, this whole structure is called an axon. Okay, just a nomenclature. And this is the shaft, and this is called a growth cone. And that also has brain here in that palm. Why? Because it's so long, it does not want to be controlled from here to here. It wants to control individually. So if there is a chemical attractant, or as I will show you, if it is a physical cue means a physical parameter like heat, like force, like fluid flow, I am going to show you, or stiffness. If it feels something, here itself, it makes decision. Right there, the signal comes through all these fingers, they get integrated and ultimately they decide where to go. Okay, that's how they program. Till now, mostly it is thought to be chemically guided. Okay, so I am not going to go. So these are called philopodia, and this is the growth cone. Okay, so what is the detail, how it happens inside? There are a lot of processes happens inside. As you will see here, even this is how the real axon looks like, the real hand of the neuron. Okay, so inside each small of them, these are hundreds of 50 nanometer thickness. Each one you are seeing. So inside those 50 nanometer, there are a lot of processes happening. And most interesting part is, if you cut one of them, that also can sense chemical cues, different drugs. Not only the whole growth cone, but also a small portion of it. A finger, if you cut, that finger can sense and move. So it's very, very computationally intensive process which goes on when one neuron finds its partners to bind. Okay? So I'm not going into more detail, but if you can imagine why this guidance is very important. Why it's important? Let's say your nerve bundle is coming and it should go to this finger, so your brain says to move here, but if the nerve, the neuron goes and connect to something in this finger, then you want to move this finger, you will move this finger. So the guidance of these axons is very, very important. If in your brain some neurons get misconnected, then you will smell blue. Okay? So you will think abrupt things, all neurological disorder. By the way, as you guys know, in Europe or United States, the cost of neurological disorders is more than the cost of diabetes, cancer, and heart attack, stroke, all combined together. Neurological disorders are growing at a very fast rate in this complex world. Okay? So what are the existing methods to guide? I have to go a little quick, I think, because I have to cover all the technology that we have. One is mechanical. People confine the axon into boundaries. Then neuron cannot move away. Okay, they have to put boundaries. But then that's not very flexible. We want to change something, let's say, in the body, or even under the microscope. We cannot just make boundaries and make cells grow like that. Okay, an electric field is also very complex to apply electrodes there, and also there are some hybrid approaches. But light can provide very good spatial accuracy and non-invasive, as you know, if we can guide with light. Okay. So our lab is fundamentally not only developing the technique, but discovering how physical cues are responsible for axonal guidance. Okay? So this axonal path finding or guidance is very important, as I told you, for an organism to process, store and retrieve information. Okay? How they are connected. Till now, only chemical cues were studied, but we are trying to study physical cues. Where we got our motivation. See, when neurons are growing, being a physicist, and you know, when in the womb, when embryogenesis is happening, neurons are developing, blood flow is happening. So blood where is flowing and neurons are also with the fluid flow. Temperature, if there is a flow, you imagine there is a temperature gradient. So there are physical cues, there is pressure, force, there is stiffness. The neuron has to go through hard surfaces, soft surfaces. So we think the physical forces are important, not just a chemical gradient, which you can imagine something happen here. So from here it can sense the chemical gradient is not feasible all the time. Chemical cues are definitely responsible, but what we hypothesize, the physical cues that I am going to describe is force. You know force. If you force the neuron, can you bend it? Flow. If you give a flow, as I told you, blood flow is happening, or some flow, if it can guide the neuron. Heat, temperature difference, can it guide the neuron? Stiffness, we prefer to go on soft track or hard track depending on different situations. Can neuron also prefer to go that way while it is going through the brain, making the circuit? You understand? So it's a fundamentally different dogma where we are asking this fundamental question. Don't confuse this with what we do when you hold a cup of tea. That of course our neurons are sensitive. Some sensory neurons are sensitive to temperature, physical cues. So like we hold this thing softly, a baby softly, another object very hardly, a hard object. If there is a temperature, then we will sense it. Then when we are drinking tea, we smell it, all this. These are sensory functions. What we are talking about is guidance when the forming connections. These two are different things. People should not confuse with that. So these are four different, I am going into more details on them because they are very important. How we are applying force on the molecules or on the neuron to make them guide like this. Or how we can use, all these are generated by light, by the way. And light is very unique. Light, you put light, you generate force. You put light and drive some motor and generate flow. Okay, and then we saw that neurons are guided by flow. You put light here, like this red spot, you generate temperature, very low temperature. You don't have to have fire there. Or you change stiffness of the substrate in which cells are growing, and then you can make them. So these are different cues that we are working on. So I will go into very quickly into this. So if this neuron is growing, you cannot imagine that you apply force and just pull it, then it will work. No, it does not work that way. If you see here, this is an object attached to one of the finger, and I am trying to pull. Like a person you want to pull with his hand, but it has its own brain. As I told you, it won't go. It will rather retract, like you see. It will recede. Why this one is pulling me? Maybe there is a range of force you can add, but the force we are applying is in piconewton, very small force. Still you cannot make it. So it retracts. Neuron has sensory system. It retracts. You want to break one of the finger, this side, and to think that it will go that side. So that also has not been very successful. So there are some videos here. But what we did is, if we align the philopodia like this, using a laser beam, you can, this is a laser beam, it's like a tunnel, and that can align the fingers into one side. Don't pull it, but push them into a side by light. If you apply that kind of force, you see here how light is pushing even the philopodia. You can push things inside cell. This video is in a very zoomed version. So you see small molecules, intracellular object are being pushed inside along a line. So if you do this kind of thing, so you are applying force inside the cell on the molecules. If you do that, then we saw that you can guide one neuronal processes to the other. Okay, and the mechanism we are studying, how it, what is the mechanism? Is it force? Is there a temperature also involved in this? We are actually looking at those right now. You can see how they are aligned inside a cell. This is a neuronal cell. You can apply with an optical fiber force and align molecules inside and even stretch a cell. You see how light is attracting it and even stretching little bit. Okay, so that optical fiber we use to guide the neuron like this. We bend it by applying force. Now moving on, after force, we wanted to see if neurons are sensitive to flow. Okay, so how to generate flow? So there are microfluidic pumps. You have to have flow at micro scale. Okay, but let's say your neuron is growing and you want to have a pump here. It's very difficult. You develop a pump and neuron axon may not be here. Its tip fingers may not be here. So what we did is we use physics by which we put some circularly polarized light. The light which is a polarization electric vector going like this, let's say clockwise. And then when it goes through any small calcite, calcite is in the ocean and everywhere, I will show you, calcite, calcium carbonate. So that has a property, if light goes with this polarization, it will change its polarization to reverse or some other direction. And because of that, light carries something called spin angular momentum. Light has also angular momentum, not only just force, but light can have torque if it's polarized that way. Because of that, the net angular momentum can be transferred to these microscopic calcite particles that you see. And because of that, they can rotate. And not only rotate, now you have a motor which is light driven, you can move it wherever you want. And that generates a flow. And see, you can control its speed with very good accuracy because light power can be controlled. See how a neuron responds to the flow that is generated by here. And that came into the cover page of Nature Photonics, where we have one motor here rotating anti-clockwise, another one rotating clockwise. Okay, I will show you video how this motor is rotating. It's a spherical motor. You can still see it's rotating, how the knob is bending like this. Okay, now you will see two motors, one rotating this way. We brought it from this end to this end. This rotating this way. You will see the knob which was going this way will now turn back into sieve through this two fluid flow channel into here. So one fluid is going this way, another fluid is generated this way. Slowly you will see the neurons will go into that. It will prefer to go between that. So it's not just a tool to guide neuron, but it first time showed that fluid flow is responsible for neuronal guidance. Okay, and my lab, my graduate student Brian, he developed something fiber optical tool by which you can generate fluid flow so that we can go deep. With a fiber, you can not only hold now an object, but also you can generate flow. You can see something is moving around. That's because of the flow. Okay, but we wanted to be sure. As I told you, light can generate heat, light can generate force. We thought might be it is possible that something else is happening. Is it pure flow? And for that, with collaboration with Dr. Jan from mechanical engineering, so we modeled how much flow will generate how much force, and we showed we generate a micro tube and we applied flow to show that neurons can be bent like this. So in this system, we not only have a laser which is driving the motor, but also these are, by the way, neurons derived from the eye of a goldfish. Okay, so that goldfish neuron is guided, and I am going to show you. It's getting published in Nature Scientific Report. It's in now proof. You can see the flow can guide and connect to another neuron here, axon here, to make circuits. So that's by pure flow. Okay. As I told you, it's not just force. There are biochemical changes happening at the growth cone level inside. There is processing happening. Not pushing something, as I told you, does not help. Just pushing. Okay. Now moving on, we saw till now how force and flow are important that we discovered to guide neurons. Now the most important one is inside the body you can give flow and things like that, but controlling flow, let's say at the microscopic level, is very difficult. Okay, it's a fundamental discovery. It's responsible while the neurons are forming circuits or migrating from different parts of the body. As you know, neurons still migrate in the adult now also, not just during development, to this olfactory system. Neurons are getting replenished now and then. So now we thought, is there a very good way by which we put light, not apply force on them, but another way by which we can guide neuron? And accidentally we discovered that if you put light in front of the neuron, not put onto the neuron, no motor, nothing, just pure light like coming out from this very low power light. we found the neurons are bent. This work is done in collaboration with Dr. Youngtae Kim in bioengineering. So you see, we put the light and we found the neurons are bending, surprisingly. So we thought, what is the mechanism? Okay, you can see here how the neuron light is fall. This is an axon which is bending here. We named it as neuronal beacon. Like a beacon guides the seed, how the neuron is guiding. We did not have the mechanism, but now you can see the laser beam then making the neuron turn this way, laser beam this side make the neuron turn that way. It's recently published in +1, and we found for one spot of laser, the neuron can be turned 51 degree, and these are again neurons from the retina. If you put the laser at 2 spots, you can turn it like this. Okay? So you can create now laser net by using like lines, crisscross, laser nets to define the path of your circuit. And this requires very low power laser. And what we recently found out, if you do a simulation, I'm not going into all these details, that how efficient it is. We found till now all the neurons which advance, 100% them by the way are affected by this cue. Till now all other methods we have been using, chemical methods, are very less effective. And this is a very controlled way, and we found the temperature rise when you use a 700 nanometer, like close to red wavelength laser, the temperature rises around 0.01 degree centigrade, 0.01 degree at this power. And at 970 nanometer, if you go towards near infrared, temperature rise is 0.5 degree. And when we guide with all these different wavelengths, what happened? We found neurons is turned for all those wavelengths. So then we thought it might not be temperature. Then what it can be from a distance? Because we thought from classical knowledge, how can neurons sense 0.1 degree? So less temperature rise. But still we cannot rule out that. I'm coming to that, how we did not rule out that. But you can see we can consistently do the guidance and turn the neuron completely 360 degree. This is by the first time, any method, not just optical, not this temperature, how you can guide an axon to 360 degree to form loops in the axon. Okay? So this one is also going to come up in Nature Scientific Report. So you can see here, with this laser, we can form loop like this, different loops of different size, radius of curvature. Okay? We can form the loop, and as I told you, this is the first method. Why it is important, this loop formation? Again, let's say your nerve, the axons from your eye is going through the exit point to your brain. So if it sees a physical constriction, it cannot go through, it may bend and try to form loop. So people have hypothesized something called that an axon cannot form loop on itself, and it is called in biology or neuroscience self-avoidance hypothesis. Means the neuron doesn't like to grow on itself. If that is the case, if it likes to grow on itself, then we will never form these connections. Or synaptogenesis, this is the formation of connection, rules out that a neuron can form connection with itself. It only happens when you have diseases like autism, retinal dysplasia, lot of diseases, genetical disorders. Okay? But we showed that if neuron obstruct a physical cue, it can form loop. Why we say it is? This is self, we call it self-fasciculation. Like in a car, you have accelerator, you have to have a brake. If you are forming connection, you don't want to have connections everywhere. So we have another hypothesis where neurons can grow on itself, and that you will see here how it is growing on itself. Mark this black point, which is laser, and these are the neurons derived from the cortex, and the laser point is fixed. The neurons are moving, forming loop and growing on itself. A zoomed version, again I can show you how the neuron goes, and constantly when the laser is in the front, it avoids it because of temperature, I am going to tell you, and it forms a loop. Okay, so this shows, this rules out the self-avoidance hypothesis. So not only the technology will be useful, what I am presenting today to make circuits. Now you can see with what precision that loop we form has radius of curvature of micron. 1 micron, 2 micron curvature. That also shows something very important, that people thought you cannot bend neurons so finely because inside there are like tubes, they will break. But we saw no, it can form radius of curvature which is very thin, 1 micron, 2 micron radius of curvature without breaking and making the neuron still grow. while it has formed the loop. So there are some physical, biological consequences to this technology that we are developing, not just technology per se. And let's say you have a loop which is forming already. Actually, we found in fact in maybe few neurons, 4 out of 600 neurons are forming the loops by itself without applying laser. That happens if we found in a dish, axon is going and there is no partner. So it decides to loop back. It does not want to go further. That we found in a dish. So if it is forming the loop, we can put the laser and then we can make it again straighten out. We can stop the loop formation. Okay, this shows the mechanism. I am not going into details, but what it shows is if we use laser of less than milliwatt at near infrared, it is equally effective as laser at 750 nanometer. So 750 nanometer laser, if you require let's say 10 milliwatt power, we can do the same thing at 1 milliwatt if you go towards near infrared. And as you know, near infrared can go deeper in the brain or into the tissue, nerves, let's say spinal cord or your eye. So it has in vivo possibilities, okay? But the most fundamental discovery is neurons can be guided by very low temperature gradient. That's most fundamental. It's not just the laser can do the technology, but the temperature is the cue now for guidance. So this shows that since temperature rise is more at these wavelengths, the effectivity is more here at low power. And then we study the mechanism, how calcium is going, we block ion channels. These are details for general public I am not discussing here. But how putting a laser at a distance from the axon can create calcium changes inside the cell by this temperature, very low temperature rise. We are studying those. But now also we want to rule out that is it real temperature effect. People say, oh, laser can generate some force, maybe this philopodia is coming, interacting and all these things. So like the fluid flow, if you remember, we said let's generate temperature without laser to confirm that this is because of laser. So what we did, again with collaboration with Dr. Youngtae Kim and Dr. Jain, so what we did is we created a micro heater, a wire and pass electrical current. And then you see the temperature rise we generated is maximum 1 degree, less than a degree centigrade, over a millimeter distance. Okay, and neurons were grown near this heater. And when the heater heats up, and you will see here, mark here, when the heater heats up in this direction, the neurons will grow back away from it. So this is a pure temperature based effect, which we can generate by light. Okay, so you can see here all statistics, all kinds of things here, the neurons bending. Now I am not going too much into this, but the last one, till now we have discussed force, flow and heat, these three that we have been working on. But also you can generate, this is a gel in which you can put some photo initiator and put the laser and create stiffness changes in the gel, means hard and soft surface. Let's say neurons are growing in a gel and we want to see how the neurons respond to that. If you see these lamps, I'll play the video. Here is the laser beam, the shining one. It is forming lines. These are not lines, means these are stiffness changes. Some property of material is changing. And we are studying, and that you can do also by using an optical fiber, like a Spider-Man. You can see it's generating these structures in the liquid Spider-Man. So light is generating these structures, the stiffness change. Then we wanted to see, we form this structure and we put in the gel. Then we put neurons or glioblastoma cells into them. If you see the structure is below this bulk of cells, the cell starts to migrate. So this has not only importance in neuroscience, but also for cancer. How, let's say, bare breast cancer cell migrate. Why it will migrate? Maybe it is affecting some physical force, then it starts to migrate near the wall or thing. So it's not just chemical cues guiding there, I want to move out. So there can be, so whatever I am presenting today has application in a lot of fields of biology, okay? So this, we showed that by stiffness we can guide neuron. Now I have to go quickly because of lack of time. So we have only covered till now the two, how to position neuron and how to guide and to connect them. Then comes laser scissors, that if you don't want how to disconnect them. See, these are networks of neuron formed. These are axons. We shine femtosecond laser here. in this mark spot, and you can see we disconnect this axon from connecting to other side at this point. It's cut. It's very precise. This is 200, 300 nanometer thickness that you can cut. Not only 200, 300, you can cut even less than that to 50 nanometer. Here if you see neurons are growing here and you cut it inside a channel and they repair if you don't cut completely. Okay? So while doing this cutting, we discovered something, that let's say you have a neuron, one neuron going here, another axon growing here. If you cut it here, see you can imagine here is thick, you made it thin. There are some chemicals released or some processes going on by which the neighboring neuron is migrated in the circuit towards it for its help. That is not shown earlier in central nervous system neuron. So people say Schwann cells migrate and all kinds of things, but this has not been shown. So that is being used. You can see again here, this neuron was growing towards right. We cut it here, you see here, and it suddenly repairs. That's another big thing we discovered. That if you cut the neuron near the hand, it repairs automatically within a minute, as compared to taking long time. You see here very quickly, 90 to 150 seconds, it's repaired. Even if one thin filament is connected, it pulls it back like a spring and connects it. It's very flexible here. That's why we can bend it so nicely. It's very soft here. That's why we could bend it, if you remember. Okay, you can again see here how it repairs within a minute from here to here. And we did the same thing in the retina. We cut it and we repair it. Okay, so this is a retina piece in which we are cutting the neuron. Okay, so I'm going to go quickly now. Now we have the circuit. I told you that we formed the circuit. I'm going to show you the circuit at the end. But you have till now shown, I have shown that how neurons can be organized by light, how they can be connected, how they can be disconnected. Now, let's say these are different parts. These neurons are there. I want to now make some particular function. I want to give a particular function, teach him something. Okay, I want to teach this neuron to fire like this. Okay, 100 hertz or 50 hertz. I want to transform that neuron. So I want to deliver a gene, a RNA molecule, something I want to deliver. How can I precisely deliver? Okay, that is done by something called laser transfection that we have developed. Okay, what are the different methods till now used for delivering genes? Is a viral method. So if I have a circuit and I put virus, what will happen? The virus will carry the gene and spread to all neurons. I don't want that. I want only to transform specific neuron in the circuit. Do you understand? So that they will have specific function. So what we do, the same laser you saw which does the surgery, we do nanosurgery, make 50 nanometer holes in the cells. This is just to show you clearly how we create arrays of holes in red blood cells, for example, without killing the blood cell. Blood cell is 6 micron and the holes are less than 100 nanometer. But we cannot resolve it unless we use AFM. So you see these spots as 1 micron because of light microscopy, but these are 100 nanometer we have seen. And if you do that hole formation, we deliver even antibody. So this has importance even in biological sciences. Okay, that how can you deliver some molecule, not just genes, which virus cannot carry antibody. An antibody generally does not go inside cell. But we can deliver, these are anti-mitochondrial antibody. In this area, we deliver this antibody into the cell. Same thing we can do to the neurons, okay? So here you see how my graduate student Kamal Dhakal, he has delivered actins, actin staining dye inside the neuron and stain them. This is in press in Nature Scientific Report again. So you can see this dye molecule has been injected, which generally does not go inside cell. And you can see how an intracellular object is moving here, okay? And we can see how the dye goes inside and how the intensity of the dye, everything change. And you can see the precision of the technique. Here are two cells very close by, a micron separated. So you don't know what these processes you see here is connected to this or this. But if we deliver this molecule to this cell, you can see which are lighting up. Do you see that? So in the network, you could not know where these things is connected, what these processes are. But you can tell now that these processes are coming from this cell, not from the neighboring this dark cell. So we have that much precision. single cell level, okay, to deliver even if the two cells are very close by. Okay. So now what we are doing with that, not only to in vitro, but we can create these small black dots in retina circuit. Let's say we derive the retina and we want to understand the retina circuit in vitro. Okay. So see, we made a triangle, so we could deliver the gene that we wanted to deliver into a triangle, rectangle, or into a periphery like this. This has a lot of importance also for in vivo disease of blindness where the vision lose from the periphery. So if we can deliver the gene by laser to the periphery, person can may be able to see. So what we deliver? We deliver the gene which makes the rhodopsin that is in our eye. So if we can deliver a gene for rhodopsin, so the cells which has lost their function towards light can be restored. Okay, so that's a complete separate work we are doing on vision restoration. I am not going into that right now. And also in collaboration with Dr. Ali Koymer in physics department, we have used not only now till now what I showed you are femtosecond laser to deliver genes, but also what we did is we put CW laser beam, few milliwatt of infrared laser beam to heat up cells more by putting some carbon nanoparticles. So if we do that, what happens? There is temperature rise happens, no holes per se, but the temperature rise stress the membrane and allows the drugs to go inside. Again, this is published recently in Nature Scientific Report, and we could deliver the gene also to the cells using this method. So here, remember, a continuous wave laser is used. So now I'm moving towards the end. The two more items, but I have 10 minutes. So one is optogenetic stimulation. This is very important to stimulate specific type of cells. As I showed you, laser can be used to form circuit, cut, delete, inject things, transform. But can we stimulate? This is a cortical neuron by light. Yes, we can. See, you put light and you stimulate the cortical neuron, and that is being seen by increase in calcium, what we are seeing. But this method is not very precise as electrode based method. You want to stimulate something in the brain or in this circuit, as I told you, it's not very uniquely specific. It can be precise in vitro, but it is not specific to particular cell type. How can, let's say you have 100 different types of cells in the brain and you shine light, how can one cell be stimulated and not the other? Because this precision I showed you, you cannot do it in the very cube volume. Okay? So Francis Crick, who got Nobel Prize for DNA structure, he imagined, without that time there was no method, this method was not there, that how can you turn on particular type of neuron. In his lecture in San Diego in 1999, he said maybe light is a tool. He did not know how it has to be used. But this technique is called now optogenetic activation. It uses opsin, not the rhodopsin in our eye, but an opsin derived from lot of organisms around the world, whether it's a desert where organisms are surviving, whether it's a pond, algae. So these also have light sensitive proteins, these organisms. So people have been studying this from 1970, but never applied to neuroscience. So in 2005, this became a big discovery. If you see top 10 discovery in that decade, it was discovered in 2000 to 2010, is when you put these opsins, this is the history of it, I am not going into detail, where if you put this opsin into the neurons, they become light sensitive. Now it is being applied to these primates also, okay, and human clinical trials will be coming for different medical applications. But how does it work? You have an algae which swims towards or away from light, and it has an eye, and that eye has a protein, as I told you, like our rhodopsin, and that protein, if you can have a DNA which codes for that protein, you can take that DNA and put into the neuron, brain, heart, cardiac, all kinds of cells. I am not discussing now, even cancer cells, and then that protein will be expressed in the neuron. These blue circles you see. Now if you shine light, these blue circles are going to open up. This protein is going to unfold or open like this, like an ion channel. And because of this unfolding, current will flow. Similar to what happens in a solar cell. Light generate voltage or current voltage. Here also you can generate voltage on the membrane. Okay, and that can control behavior and things. Okay, so the intensity required for this is very low. So you can use a micro LED, and since it's a genetic technology, you can deliver to whatever gene cell type you want to deliver. Let's say you have 100 different types of cells, you can deliver to one type. So there are different advantages. I am not going into details of that because of lack of time. This is less susceptible to contamination. You don't have to put hundreds, thousands of electrodes like you put in the retina to stimulate. So in collaboration with Dr. Chiao in electrical engineering, we have developed micro LED and electrode array by which we can shine. This is very thin, less than a millimeter thickness, like we can put into the brain and it will shine light and you can control activity. And not only that, you can record electrical. But as you will see, this also damages little bit brain because you are poking something. You are using a blue light for this optogenetics, and blue light cannot go deep into the brain. So in first time 2008, I showed that you can use infrared light to go deeper and more precise stimulation to stimulate particular single cell resolution. That's what is shown in this paper, Biophysical Journal. So you can stimulate with single cell resolution. My student now developed fiber optic method of that so that you can go deeper. And we did that also in vivo, and the paper is in minor revision, that we could put into the, without opening the skull or the dura, we can stimulate deep brain structures and still get neural signals. So we don't have to poke fibers inside. So this is also done with collaboration with psychology professor Dr. Linda Perotti, who helped us in, you can see here how the laser is opening here and that time neuron is firing. So we can synchronize the neuron, tones, music, you can feed into the neuron. Whatever tone you want to, like a piano, you can make the neurons fire as you desire, not how they desire. So you can keep on doing that, and neurons acquire that property. Okay, then you can see how they do that. Okay, so we studied in more detail, but you can see this is in deep brain region that we can stimulate with the red light instead of the blue light. So this is done with collaboration, as I said, in psychology. So now we are studying how pan circuitry of brain works. Earlier we were using blue fiber and making little bit hole, few millimeter into the brain. Now we can do it by infrared without going through the dura. So you can see if you use blue light, you damage the brain little bit because you are inserting a fiber or the LED, micro LED, but with 2 photons there is no damage to the brain. Okay. So now coming to the last part, but I don't have time, but this is the most important one also. We got NIH funded for this, is how to do optical detection of this neural activity. That we want to detect optically, because if you want to poke electrode everywhere, the cell will be destroyed. You cannot sustain the cells or the circuit for long term. So we want to detect it optically. So there are 3-4 different methods we are using. NIH funded us for particular one method. So this is how you record electrically. You poke electrode to record its activity, or as I showed you here, these are electrical activity we recorded using the device in collaboration with Dr. Chiao, that we could record how neurons stored information, things like that. But optically, if you can detect, for example here, you can use a calcium dye to see, but calcium dye can also lead to long-term toxicity. Okay, this is a fluorescence method. So we want a non-fluorescence method. This is, I am not going into detail, but this actually makes where we have used lasers to deliver the gene into the neuron, specific neuron, and use the 2 photon I described, infrared laser, to specific layer stimulation and detect also by calcium dye. This is how we are probing neural circuitry. Due to lack of time, I am not going into this. Also, we can make calcium release not only from single cell, but inside the cell, like organelles. We can make so precise stimulation. But here is a circuit. I want to show where we have make a circuit. As I showed you, these are 2 cortical neurons, okay, from the red cortex, and this is astrocyte. So we are probing now communication not only between neurons, but also between neuron and astrocyte. It's very important. Till recently, people thought a lot of our brain portion is covered with astrocytes. But people thought astrocyte does not do anything to the neuronal function. But now it has been discovered it has a big role to play in different diseases, different signaling processes. So now we form the circuit, and these calciums, we did not put calcium dye now. We did genetically encoded voltage indicator. Means if they will fire, calcium will increase automatically. So they are not with dye. So, and we stimulated with laser. So you see here, this is one cortical neuron, another cortical neuron. These are astrocytes and they are connected. Okay. What we found, if you see the video of this one, we stimulated laser here and the calcium, how they are communicating. We are studying how these signals which are coming out, the profile you are seeing, how they are delayed, whether it is coming through this path, like you see this path, or it is going directly through this path, with which path this signaling is happening. Okay, I cannot show you more data on this circuit because they are in other journals, but this is the optical detection part for which NIH funded us. We hypothesize, can we detect without putting any dye or genetical indicator, if there is ions moving inside cell, is there a mechanical deformation happening? And how much the thickness is happening? Can we detect that? It may be sub-nanometer, but there are optical methods, interferometry method. If you see here, if you put blue light, so we hypothesize the neurons will swell. This swelling is not like this, but swelling will be in nanometer scale. Okay, and that one can we detect? So we developed something called optical coherence tomography used for retina scan, but this is a most sensitive one where you can get picometer resolution. If you see here, the calibration, the noise floor is 80 picometer. You know, atoms are how much? Angstrom. Very close to very atomic resolution. So you go to 80 picometer, okay, and then we found if you stimulate at particular 30 Hz frequency, we can detect that signal. This is without dye, this is not with electrodes, okay. Then, but this method can detect at a point, then you have to scan to see. So to develop a non-scanning method and label-free method, we are working on new technology which we have been using for different other products, but same thing, using optical interferometry, changing the phase of light. Okay, so this is in collaboration with a startup company we have, nanoscope technology, in which we could see nanoscale fluctuation in cell, okay, of 1.4 nanometer resolution. This is a cell which is rotating, you can see how the intensity is changing. Okay, and that one we combined with this optogenetic stimulation to show, you can see here, the resolution is 1.4 nanometer. Okay, the noise of this instrument, but we can see the cells completely. You don't see point by point. This is a cell with the opsin I talked to you. Okay, that's why it is fluorescent, it's not a dye. So if you stimulate these two cells, one cell shows more fluctuation than the other cell. So we believe this can be a future technology by which this is not a dye color, this is a pseudo color of light. And also we are using polarization of light to detect. This is another method by which light you put, when you stimulate the neuron, there are some membrane changes happening and that will change light polarization. So that's another method. Okay, so to summarize, the most important message, the physical cues such as force, flow, heat and stiffness and stimulation can be highly efficient in guiding. I did not describe stimulation. Stimulation by means, let's say you are going in the car, you are listening to music, same thing, neuron is growing and it wants to find its target. If we can stimulate, encode it somewhere that it goes to the muscle, then to the sensory organ. Can we encode something, okay, so that we can direct its path by encoding some information into the neuron, go this way or that way. Okay, so our integrated optical approach to understand neuronal function, use the technology like I showed you, the guidance, functionalizing neuron, encoding desired neural activity, whichever frequency we want, whichever tone we want, deciphering the code in a label-free manner, and the understanding of the mechanism by which a single neuron sends and processes information in a circuit, how it contributes to the circuit is very, very important. The most important thing is how it does cognitive function, is diversity among neurons. Unlike the von Neumann architecture in the computation, where every element is very well defined, the neurons from a particular regions are diverse. Because of the diversity, they can generate this system which does cognitive function. So we want to find out those neurons, put them together, see what is the number of neurons, what is the diversity required to generate these emergent states which creates this decision making, knowledge out of knowledge. So these are the fundamental questions. How And this, if we understand, we can not only control migration, I showed you, activity, but also differentiation of neurons, like stem cells. Neural stem cells, if you stimulate them at different frequency, different tone, or a different substrate, which we are collaborating with Dr. Yongtae Kim again, and we have shown some result. If you give different frequency of light, we can make them differentiate into different type of cells, okay, or repair, we can enhance. So these are the challenges, but at the same time, lot of students, now my new postdoc also has contributed to work, Subrata and new graduate student from electrical engineering, Sarmista, and there are a lot of undergrads who are doing very good work. I don't have the pictures right now, but Brian Black is a graduate student who worked mostly on axonal guidance, who did very good work. Kamal is working on optogenetics, and I have a long list of collaborators, present and past, and we are thankful to UT Arlington, President and the Provost, for the grants given to a lot of this work, and also the funding from NSF and NIH, and also instrument grants. Instruments were provided by local company Plexon and Nanoscope Technology for imaging and neural recording. So I thank all of them, and thank you all for your attention.